Lucy! Lucy! DNA is found inside the nucleus, which is the center of a cell. Cell. Pause. DNA. Here. The different nucleotides of DNA that pair with each other are. These are both strands of DNA. When there are two strands of DNA, A pairs with T, G pairs with C, and then it could also be the other way around. But when the body is trying to make protein, instead of two strands of DNA, there's a strand of DNA and a strand of RNA. When DNA is pairing with RNA, it's pretty much the same thing, except for RNA isn't able to make T's. So when it's an RNA... Hello, stop. Wait, no, So, when DNA is pairing with RNA, it's pretty much the same thing, except for RNA can't make T's. So instead it has to make a U to pair with the A. Although RNA can make A's. So, yeah. Then the C would pair with the G, and the G would also pair with the C. Now we're going to talk about DNA mutations. A mutation happens when... No. A mutation is when... Stop, Khalil. So, a mutation happens when a radiation, like X-rays, UV nuclear radiation, or something I don't know how to pronounce, is like carcinogens, which are like toxins, the stuff in cigarettes and other things, go into your body, and they basically picture this T would get changed magically into an A. And then instead of the question mark being here, this one would get changed, this one. So since that was a T originally, since this is RNA, this was a U, but then it also got changed because it needed to be able to pair with the A. So stop yelling, guys! Stop yelling! No, this one was A and this was RNA, and RNA can't... Stop yelling! And RNA can't make T's. Stop it! I'm trying to work, please. Please! I'm sorry. Then this would be then changed into a U. So then there would be a different... Like, let's say these three letters, they don't, but let's say they accidentally made stop, because all three of them, each three... I'm trying to record. I'm moving. Wait, no. Okay, so sorry I got away from them. Now I can focus. Oh, Taylor. So when the radiation or the other thing came along and changed the T into an A, this also, it was an A. It also changed this and turned it into a U, so the U could match with A. Usually a T would go with A, but since this is RNA, RNAs aren't able to make T's, so they have to make U's. Earlier, these three nucleotides, every three nucleotides lead to one sequence. It could be stop or LIS. I don't know the actual names, but those are just the abbreviations. And I don't know what these three stand for, but pretend they stood for stop, which means the end of the DNA and RNA sequence. If this said stop, then it would stop early, and it would eventually lead to a smaller protein, and all the amino acids, or no, it would lead to a smaller to a different amino acid mixed with all the other proteins, which would then lead to a smaller protein since it stopped early. Or if it were any other, thing like pro or ala, then it would be a different kind of protein, and that's how the mutation in the protein is made. DNA mutation creates different proteins from the normal sequence, this is how it happens. Let's pretend a normal DNA sequence would be, a regular DNA sequence would be, let's pretend it would be so DNA always starts out with the same sequence. The same sequence is MET and is AUG. So as I said, every 3 nucleotides is a different, I forget what it's called. Oh, it's a different code for an amino acid. So let's pretend it was AUG, UUU, or CG. Oh wait, no. And then it ends with stop. So at the end, it would actually be UGA. Sorry, that's kind of hard. After that, the RNA would come and pair with it. It would be U, A. Oh no, sorry. If this was the RNA, this is the DNA. It would be T, A, C. Then it would be A, and then that would be A, C, T. That sequence would be MET. And if this is the RNA, and this is the DNA, you usually read MET, then it would be FE, then it would be stop, because you always read from the RNA. Yeah, you always read from the RNA. Sorry. Here's a sequence where I changed one of these. Instead of this being U, it's a C. As I said earlier, the changes can happen if some kind of thing like a radiation comes and messes them up. So now the whole sequence would change. Say this was DNA. Wait. This is RNA, sorry. Wait. Usually I would write the DNA first, but I kind of forgot. So this is the RNA, and usually you would have the DNA first. So it would be... Wait, no. Wait, did I say anything? This is the RNA. Usually you would find the RNA after you find the DNA. I kind of forgot to do that though. So just pretend that the DNA was here first, okay? Because I don't like clipping any of my mess ups out of the video, because then I might actually accidentally clip one of the things that I wanted to do. So, since I made this change, and this change, it will be a total change in the different sequences, and that would be the mutation. Also, the code for the amino acids would change also. This one is also MET, instead, because once the code for the amino acids changes because of this, instead of this being Fe, it's ARG. So the whole protein changes. Oh, plus all of this happens in the ribosomes. Khalil, be quiet. So this is how they're transcribed to DNA. Let's pretend that this is a normal double helix of DNA. So 2 strands of DNA. Pretend this is a normal strand of DNA, right? Let's pretend these 2 proteins come along and separate the DNA into 2 strands. Sorry, that was really messy. I did it again. Pretend the green is the protein.
I just have to add one picture.
No, I have to stop. After there are 2 strands of DNA, one of the strands is just there. Then a strand of RNA comes in. But the RNA that comes in has to be the one that pairs. Then these 3 RNA pair, code for different RNA. Code for different sequences, such as like cis, sur, pro, ala, stop, all those different things. Pretend that this is a strand with these different nucleotides. Then these little things come along. Oops. These pair with that. These together create a certain sequence, as I said before, like stop, ala, pro, and that those three make one amino acid. So pretend that this is amino acid. Then another one will come along. And that one codes for another amino acid. This connects to that. And this one just flies away and goes somewhere else, because it's not needed anymore. And that its amino acid stays to that one. And eventually, there's a long strand of amino acids connected to one of these. I didn't mean to erase one of these. Made a mistake on this first one where I was talking about how the amino acids are made. This shouldn't be a U, it should be a T. Now there are a lot of amino acids, and it's ready to make a protein. Different amino acids in different shapes and sizes according to what the DNA sequence is. That creates like stop, or ala, or pro, or sur, or ser, sur, whatever, I don't know. So then those amino acids, I accidentally deleted them. The amino acids go together to make a protein. Protein. However, if there's a mutation, the protein that can be made will be lacking in something, or it could have some kind of weird extra thing that doesn't serve its purpose. The different proteins help you with different things. They help you with digesting broken down particles that your body no longer needs, or messenger proteins, or proteins that digest. I'm sorry, but there could be, if the protein's a mutation, that protein could be lacking in something, and it might mess up its job or something like that, which is cause something good or bad to happen in your body, or in your system. Yeah. But it could be good. Like, that's how evolution happened, and how we grew to have different traits. But it could also be bad, and it could prevent you from doing certain things. The end. Sorry that I messed up a bunch of times and I couldn't delete it. But I didn't want to risk accidentally deleting any of the good things. And if I had a chance and if I had time, I would do it again and make it better. So sorry about that.
Made with Doodlecast Pro.